Welcome to the video Mitochondria. Mitochondria. In this video, we will learn the discovery, structure, and functions of mitochondria, which are known as powerhouses of the cell. And in the cell, these are the small granular or filamentous bodies which are present in the cytoplasm of eukaryotic cell. These are absent in bacteria, some algae, mature mammal, red blood cells, and viruses. Now let us see the discovery of mitochondria. First observed by Collier as granular structures in the striated muscles. Fleming named them as phyla. Richard Altman named them as bioblast. The name mitochondria was coined by Carl Benda. Michaelis used the supravital stain Janus green as a vital dye. And this is the picture of mitochondria. And here, mito means thread, chondrion means granule. It's a double membranous bound organization, energy converting organelle present in all eukaryotic cells, sites of aerobic respiration. Semi-autonomous organelles because they have their own genetic materials. You observe the picture here. And this is the picture of how mitochondria divides. The younger mitochondria localize the cell periphery. Its asymmetric division it shows. It divides by binary fusion like bacteria, self-replicating like bacteria. And this is the structure of mitochondria. And first, morphology. The size 0.05 to 1 nanometer in diameter. The length 1 to 10 micrometers in long. Shape, bean shape. In fibroblast, it is elongated and thread-like. Number, it varies from cell to cell. Example in rat liver cell, they are few to 500. In sea urchins, they may from 13,000 to 14,000. Location, cell with high energy requirements. Example, sperm cell, muscle cell, etc. They are usually located. Now let us see the structure of mitochondria. This is outer membrane, this is inner membrane, and the foldings are called cristae, and the space is between the two membranes is intermembrane space or mitochondrial space, and this is matrix. The outer membrane is a simple phospholipid bilayer, fairly smooth. It encloses the mitochondria, containing protein structures called porins. Porins allows the free passage for various molecules into the intermembrane space of the mitochondria. The second membrane is inner membrane. It is freely permeable only to oxygen, carbon dioxide and water. Inner membrane is convoluted, forming folds called cristae, impermeable to many solutes due to high content of phospholipid called cardiolipin. The cristae generally increases the inner membrane surface area. Intermembrane space or perimitochondrial space. The space between inner membrane and outer membrane. It has high proton concentration. Proteins present participate in ATP synthesis. This is the electron microphotograph of mitochondria. The matrix, it's a gel-like consistency. Dense, homogeneous. 2/3 of total protein of mitochondria is matrix. Mitochondria have enzymes, ribosomes, DNA, mRNA, granules, fibrils, tubules like these substances. Major enzymes include enzymes involved in synthesis of nucleic acid and proteins, fatty acid oxidation. This is TC cycle. means tricarboxylic acid cycle, where succinic dehydrogenase is not involved. The foldings are called cristae. Inner membrane is thrown up into a series of folds called cristae. They are called tubuli or microvilli in plants, which expand its surface area, enhancing its ability to produce ATP. Cristae is covered with the inner membrane spheres called stalked particles or knobs or heads. And mitochondria has its own DNA, which is called mDNA or mitochondrial DNA. It has 37 genes. Inner layer is similar in composition to bacteria. MitoDNA is structurally similar to bacterial DNA. Ribosomes, enzymes and transport systems are similar to bacteria. This is same size as bacteria. You can observe the picture of mitochondrial DNA, which is circular. And here the mitochondrial DNA is small, double-stranded, covalently closed, circular molecule. It is made up of one heavy strand and one light strand. Occurs in multiple copies. It has 16,569 base pairs. Most usually remains attached to inner mitochondrial membrane, stores biological info required for growth and multiplication of mitochondria, can undergo replication and duplication, and it is different from that of nuclear DNA. The next is functions. Energy transducer of the cell. This is synthesis of ATP. Krebs cycle in matrix electron transport system. This is phosphorylation, where ATP ACE is involved. Storage and transport of ATP. The ATP that are produced as a result of cellular respiration are liberated through a transporter called adenine nucleotide translocase. Enzymes required for the synthesis of lipids are present in the mitochondria. Production of heat, non-shivering thermogenesis. And here, the functions of mitochondria. The main function is glycolysis, where a glucose molecule is converted into 2 pyruvate acids, which are 3 carbon compounds. This is a cytoplasmic pathway. It occurs both in eukaryotes and prokaryotes in cytoplasm. And oxidative phosphorylation is another function of mitochondria. Here you can observe inner mitochondrial membrane where complex 1 to 4 complexes are located, and in addition ATP is there. Oxidative photophosphorylation. The process is first described by Peter Mitchell in 1961. This is called chemiosmotic theory. He postulates that the two processes are coupled by a proton gradient across the inner mitochondrial membrane, so that the proton motive force caused by the electrochemical potential difference, that is negative on the matrix side, drives the mechanism of ATP. ATP synthase uses the energy of the proton gradient to synthesize ATP from adenosine diphosphate, where inorganic phosphate is added. This is based on chemiosmotic theory, where ATP is called energy currency of the cell. A stands adenosine, T tri P phosphate. ATP means adenosine triphosphate.